What we did is we started with nanomachines. We've been investigating nanomachines for several years, particularly nano cars, which we've made previously, where we had 4 wheels and a chassis, and then we built motorized nano cars that had these little motors that could push them along a surface. And more recently, what we did is we built something analogous to a nano submarine, where we could have the motor rotate when we shine a light on it, and the motor rotates at 2 to 3 million rotations per second when a light shines on it, and we could propel it in solution. Now, these molecular motors and the molecular nanomachines are really quite small. We can park about 50,000 of them across the diameter of a human hair. And so what we've done in this study is we've shown that we can drill into cells, human cells or animal cells, we can drill into them using these nanomachines. So the nanomachines will attach to the surface, and then a light will be shown upon them, and they will drill right into the cell.
As a microscopist, the resolution of anything which we can see is limited by the light which we use to excite and to visualize things, and the optical quality, which we call the refractive index and the numerical aperture of the system, which is a constant number. And in this case, what we use in our microscope, we are capable to see things in resolution of 60 by 60 nanometer, which is incredibly small. And what allows us you to see in the video that there are 2 cells which are having surrounded by these nanomachines. So when you look at the cells, you see this wonderful green network, which is the mitochondrial network, and the UV excitation. And this ultraviolet light actually also excites the nanomachine. So as we see the video, these nanomachines are drilling to the cell, drilling small holes, allowing this dye, propidium iodide, to get incorporated into the cells that binds to the fragmented DNAs, which is one of the signs of necrotic cell death, which means the cell there's no point in turning back. The cell is already dead.
For many years, I never had envisioned the nanomachines being used medically. I thought they were way too small because they're much, much smaller than a cell. In other words, it would take thousands of these to go around the circumference of a cell. But now this work has really changed my thoughts on this, and I think that therapeutically this will be a whole new way to treat patients using nanomachines, and this is going to be an excellent application for cancer treatment and much broader, not just for the killing of cells, but for the treatment of cells, being able to interact with the human body using molecular machines.